Assalamu alaikum everyone. I am Dr. Haris Ahmed Khan and today I will be discussing in silico primer designing. In silico primer designing is a fundamental concept in molecular biology, bioinformatics and modern genetic research. In this lecture, we will not only understand how primers are designed computationally, but also will explore the scientific principle behind their efficiency and specificity. Let me begin with the basic concept. In molecular biology, most techniques such as PCR, sequencing, and cloning rely on DNA amplification. This amplification cannot occur without primers. Primers are nothing but a short oligonucleotide sequences that bind to a specific DNA region and provide a free 3' hydroxyl group for DNA polymerase to initiate synthesis. So let me draw it for you. So here we have a DNA strand. This is the 3' end, this is the 5' end, and our primer should bind here. So for the primer, 5' end is here and 3' end is here. So here it will provide a hydroxyl group for the polymerase to start the process of polymerization. So if these primers are not designed correctly, what would happen? They may bind to multiple regions or may not bind at all, or they may form unwanted structures. This leads to failed or misleading results. That is why primer designing is not just a step, it's a critical determinant of experimental success. Primers are required for PCR, and PCR is a polymerase chain reaction which make millions of copies of DNA. PCR is a common technique which is commonly used in molecular biology labs, where a small amount of DNA is used to make copies, and these copies can be used to identify, detect infectious organisms, and genetic variations among individuals. PCR was invented by Carey Mullis. He was an American biochemist and was awarded a Nobel Prize for this discovery in 1993 for the invention of PCR. So PCR constitutes three steps, which are denaturation, annealing, and extension. Otherwise, these steps may be pre-denaturation, then denaturation, then annealing, and then extension, final extension, and then is storage. So the denaturation occurs at 94 degree. This temperature can vary from 92 to 98 degree. Denaturation is important because it converts the double-stranded DNA into single-stranded DNA, or in other words, it separates the both strands of DNA and allows the primer to find their position and get attached there during the annealing step, where temperature comes down around 50 to 60 degrees. And the polymerization starts at 72 degrees centigrade, where nucleotides are added to the growing strand of DNA. So here we have a double-stranded DNA, and this is our target region. When temperature is higher at 92 or 98 degree, both strands separates and becomes single-stranded. After that, the temperature comes down around 50 to 60 degrees and the primer attached based on their melting temperature. So here we have a forward primer and this is a reverse primer. Following that, the temperature goes to 72 degrees centigrade where the polymerase starts polymerizing the new strand of the DNA. So ultimately we have two copies of DNA. So this is the first round of PCR amplification and this steps repeats for 30 cycles. During the second round, the DNA strand separates. Again, the primer bind at their complementary locations and polymerase starts polymerizing the new strand of DNA. So ultimately, we have four copies of DNA strands. So this goes like this. During the cycle 1, we have synthesized two strands. Ultimately, after 30 cycles, we have this many number of strands. So here we have a scenario where we have amplified a sequence of DNA and this is the region which is highlighted here and you can see the amplicon on the agarose gel. So as this region was known, we know the exact size of this region and here we can estimate that this was around 700 base pair. For PCR, we normally use positive and negative controls to check the process to make sure that there was no contamination during our process of experiment. So as an example, here we have a double-stranded DNA and when temperature reaches 95 degree, it separates into single-stranded. At annealing temperature, the primer binds to their complementary sites and at 72 degrees centigrade, the polymerase comes and utilizes the 3' hydroxyl group of the primer and start the polymerization process. And this process repeats again and again. Temperature goes higher to 95 degree and then comes to annealing temperature for the binding of primer and then at 72 degrees centigrade, polymerase start the process of polymerization. and this will continue for 30 number of cycles. After each cycle, the number of copies doubles, and after the end of 30 cycles, million of copies of the DNA are produced. Firstly, let's talk about what is in silico primer designing. The term in silico refers to computational process carried out using softwares and logarithms. In silico PCR designing involves inputting a DNA sequence into a computational tool, applying defined parameters such as length, melting temperature, and GC content. And the third is generating optimized primer pairs. This method allows us to simulate experimental conditions before performing actual lab work. It significantly reduces trial and error and improves reliability. So, do you know the importance of primer design? Let's understand why primer design is so important. A well designed primer ensures high specificity, binds only to the target sequence, high efficiency enables proper amplification, and high reproducibility give consistent results. On the other hand, poor primer design can cause non-specific bands in PCR, primer-dimer formation, and low amplification yield. Such errors can lead to incorrect conclusions, which is why primer designing must be done carefully. So the first parameter is the primer length. Now let's discuss primer length in more detail. Primers are usually 18 to 25 nucleotides long. This range is considered optimal because short primers less than 18 bases may bind to multiple locations due to low specificity, and long primers more than 25 bases increase binding specificity but reduces annealing efficiency. Additionally, longer primers may form secondary structure more easily. Therefore, an optimal balance between specificity and efficiency is required. The second factor is melting temperature. Melting temperature is one of the most critical parameters. It is defined as temperature at which 50% of the primer template duplex dissociates. In practical terms, if Tm, that is melting temperature, is too low, primer binds weakly, and if melting temperature is too high, primers may not bind efficiently. Melting temperature should be between 50 to 65 degrees. Forward and reverse primers should have similar melting temperature. This ensures that both primers anneal simultaneously during PCR cycles. GC contents play a major role in primer stability. Guanine and cytosine form three hydrogen bonds compared to two in adenine and thymine pairs. This means high GC content increases stability, but excessive GC residues can lead to strong secondary structures. Therefore, ideal GC content is 40 to 60%. This range provides a balance between stability and flexibility. So how would you calculate the melting temperature of a primer? So we have a formula 2A plus T plus 4G plus C. By using this formula, you can manually calculate the melting temperature of the primers, or you can use the online available web servers or tools for calculating the melting temperature of your primers. Now let's focus on very important concept, the GC clamp. The 3' end of the primer is critical because DNA polymerase extends from this end. Having 1 to 2 guanine or cytosine bases at the 3' end enhances binding stability or improves extension efficiency. However, too many GC bases at the 3' end can increase non-specific binding. So again, balance is the key. The GC content percentage of the total primer should be 40 to 60%. This is where many students make mistakes. Primer can interact with themselves or with each other. Hairpin loops, forming hairpin loops, primer dimers, cross dimers, or repeats. So for example, if a primer forms a strong hairpin, it may fold into itself instead of binding to the DNA template as seen in this picture. So this primer is not available for the complementary site and it cannot bind to that site. This reduces PCR efficiency. So here we have an example of primer dimer where both primers are binding to each other and they will not bind to their complementary site on the DNA template. So a repeat is a dinucleotide occurring many times consecutively. For example, here we have a sequence that is 80, 80, 80, 80. Maximum acceptable numbers of dinucleotide repeats is 4. And if this number exceeds, the primer may bind to itself and it will inhibit our reaction. Primer with long runs or single bases should generally be avoided. For example, in this sequence, we have a number of Gs, and if we count like 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So this number is not acceptable. So the maximum number or the value which is acceptable should be 4 or 5. Otherwise, the GC content value would be very high and it would be difficult and it would be very difficult for the primer to bind to its complementary site. 3' and stability is very important. It is the maximum delta G value of the 5 bases from 3' end. An unstable 3' end will result in less false priming. And a very important factor is to avoid template secondary structure. The stability of the template secondary structure depends largely on their free energy and melting temperature. If a primer is designed on a secondary structure which is stable even above the annealing temperatures, the primers are unable to bind to the template. Hence, it is important to design primers in the regions of template that do not form a stable secondary structure. You should also avoid cross homology, which means that the primer are designed in such a way that the sequence of primer must not amplify other genes or the primer should not bind to other regions of the DNA. So it should be specific to the portion from where you are generating the amplicon. Commonly, primers are designed and then blasted to test the specificity. Now let's move into a slightly advanced concept, that is thermodynamics. Primer binding is governed by Gibbs free energy denoted by delta G. Negative delta G indicates stable binding. Extremely negative delta G suggests strong secondary structures. And what we aim for? Moderate delta G values, stable priming template binding without unwanted interactions, thermodynamic balance ensures efficient and specific amplification. So what are the parameters for designing primer pairs? The first vector is amplicon length. Amplicon length is dictated by experimental goals. For real-time PCR, the target length of our amplicon is closer to 100 base pairs. And for standard PCR, it is ideally kept around 500 base pairs. So the product length should be equal to position of antisense primer minus position of sense primer plus 1. Primers can be located near the 5' terminal region and the 3' terminal regions. So what should be the position of our product? Primer can be located near the 5' terminal region or the 3' terminal region or anywhere within the specified length. Generally, the sequence close to 3' end is known with greater confidence and hence preferred more frequently. The third factor is the melting temperature of our product. So as already told, melting temperature is the temperature at which half of the DNA duplex will dissociate and become single-stranded. So how the optimum annealing temperature is calculated? So we have a formula here that is zero point 0.3 into Tm of the primer plus 0.7 into the Tm of the product minus 14.9, where Tm of primer is the melting temperature of primer template pair and Tm of the product is the melting temperature of the PCR product. Now let's dive into the primer design using different softwares. So first of all, let me tell you the complete workflow step by step. Firstly, you have to retrieve the target DNA sequence from any database. Then you will select the region of interest. And thirdly, you have to input the sequence into primer design software. That software will generate candidate primers and other parameters for you. Now you have to check the specificity using BLAST. You also have to analyze the secondary structure of those primer pairs. After that, you have to select the best primer pair which shows ideal properties. And in this workflow, each step is essential for ensuring accuracy. So the first step is to obtain the DNA sequence. This is usually done from biological databases. The accuracy of the sequence is very important because any error in the sequence will result in incorrect primer design. Thus, always verify the sequence before proceeding. Now let us discuss the tools used. Modern bioinformatic tools allow automated primer design. For example, Primer 3, NCBI Primer Blast, and OligoCalc. These tools apply all design parameters and generate multiple primer options. Furthermore, they also provide detailed analysis. They make the process faster and more reliable. So here we have an online tool which is Primer 3 Plus. You can paste your sequence here or upload your sequence file. Once you are done, then you need to click on Pick Primers. After that, this type of page will appear and it will design both forward and reverse primer from the provided sequence for you. The other one is SnapGene Viewer. You can download the trial version from the website. When after installing, it will look like this on your computer. You can put your sequence file in this tool and then proceed with the primer designing and it will show your primers like this. And the other one is BB Server. So on the website of Baby Server, you will have a home page like this, and you have to click on primer design. So from here, you can also copy paste or upload your file for primer designing. The other one is Pearl Primer. It is available freely online and you can download from here. After downloading, you can install on your computer and graphical user interface of this tool look like this. So you have to provide your sequence here and then you need to pick primers and it will show your primer pairs like here. After that, we have a primer quest and the user interface is very friendly. You can paste your sequence here and proceed with the primer designing and it will show your primer sets like this. So here you have the complete DNA or the region of DNA which you provided to this tool and it has designed the primer pairs as shown here. If you click on those locations, then you can see your primer pairs highlighted here. For designing primers for real-time PCR, you need to come on real-time design software. So you can download this software on your system and can make primers for your real-time PCR experiment. We also have a primer blast which is freely available on the website of NCBI. So for primer designing, you can also use this tool as well. So after coming on this, you need to click on this option that is pick primers and you are redirected to a page which looks like this. Here is the accession number and below is the option where you have to click for picking primers. So after the tool has performed the analysis, you will see your primer pairs like this. So based on the property of these primers, you can select any of the two primers for the amplification of your amplicon. You can also see the properties of the primer from here. So this was all about designing in silico primers, and this method saves time compared to manual design, reduces experimental errors and improves specificity and efficiency. But the limitations are that the software predictions are not always perfect. Experimental validation is still required. So if I conclude, in silico primer designing is a powerful integration of bioinformatics and molecular biology. It allows us to design precise and efficient primers, reducing failure rates and improving experimental outcomes. Understanding both theoretical principles and practical tools is essential for every molecular biologist. Thank you for your attention. I hope this lecture helped you understand both the concepts and scientific reasoning behind primer designing. I will be very happy to take any questions.